So students, today we are going to study about the entry of fructose, mannose and galactose in glycolysis. Now how these sugars they enter into the glycolysis. Now we are all familiar that whatever we eat, we do not know what type of sugar is going inside the body, because many of the food content they also have glucose along with the fructose. Some of the fruits they also have only the fructose. So in this case, what happens to the fructose, mannose or galactose in the body? Now see, uh, glucose is the main metabolic fuel in most organisms. So we are all assured that glucose is the main metabolic fuel in most of the organisms. But there are other sugars which are converted into the glycolytic intermediates such as fructose and sucrose. Now we know that sucrose is converted into fructose, are the major sweeteners which are present in the foods and beverages. Likewise, the lactose, which is from the milk lactose, a disaccharide, that is galactose, is also present. Then mannose from the dietary polysaccharides and the glycoproteins. So these are the various sources of the mannose, galactose and fructose. We have seen the glycolytic sequence, whole 11 pathways which contribute to the glycolysis. Now we have to see, although we know that these are not the, see the structure of glucose. Now in the structure of glucose, what is the difference between the structure of glucose, the structure of fructose, the structure of mannose and the structure of galactose? If we see at this, we come to know that in case of fructose, what is the difference? Here we have the aldehyde group, while we have in the fructose, we have the alcoholic group. Okay. Now in case of mannose, again we have the aldehyde group over here and OH group here, which is not present in case of glucose. In case of galactose, this group is similar in the three sugars, but here you can find that the position of the OH group, here the OH group is there, which is not present in glucose, and again one OH group is here, which is here and which is not there in glucose. So the with the clarity in the OH group, whether it is present in the with the carbon atom, we can say the structure is decided. So now how do this particular other sugars they enter into the glycolysis? So first of all, we will see how the fructose enter into glycolysis. So now much of the ingested fructose is metabolized by the liver. So whatever amount of fructose we take from the food, that is metabolized in the liver. It goes into the liver. Now in the liver uses the enzyme for fructokinase, and this fructose in the liver is converted into fructose 1-phosphate. So this is the first step in the phosphorylation of fructose to fructose 1-phosphate by the enzyme fructokinase. Now here we can find that fructose is converted into fructose 1-phosphate, and here the enzyme is a kinase, and we know that the kinase enzymes they require the ATP. So this ATP is converted into ATP by donation of its phosphate group to fructose, so that fructose is converted into fructose 1-phosphate. So this is the first step. Now this fructose 1-phosphate is then split into glyceraldehyde and dihydroxyacetone phosphate. So we get the two molecules. one of glyceraldehyde and the other is of dihydroxyacetone phosphate. Now the enzyme here is the aldolase, which converts fructose 1 phosphate to glyceraldehyde and dihydroxyacetone phosphate. Now this glyceraldehyde is then again phosphorylated to glyceraldehyde 3 phosphate. So here we get a molecule of glyceraldehyde 3 phosphate, a glycolytic. Now this we know that this glyceraldehyde 3 phosphate is a glycolytic intermediate. So this is the glycolytic intermediate. Now further this glyceraldehyde 3 phosphate is now channelized into the glycolysis. So fructose is converted into glyceraldehyde 3 phosphate before it is going into the glycolysis. So see, these are the structures. Fructose is converted into fructose 1 phosphate. Here is the phosphate group. This fructose 1 phosphate is split into two molecules, one of glyceraldehyde and the other of dihydroxyacetone phosphate. This dihydroxyacetone phosphate undergoes isomerization and is converted into glyceraldehyde 3 phosphate, and the enzyme is triose phosphate isomerase. While this glyceraldehyde is converted into glyceraldehyde 3 phosphate. So this is the overall reaction. Now after this point, we know that all the glycolytic sequences are same as that of the glycolysis. So fructose has to enter the glycolysis like this. From fructose to fructose 1 phosphate, fructose 1 phosphate to two molecules, one of dihydroxyacetone phosphate, another of glyceraldehyde 3 phosphate. Glyceraldehyde 3 phosphate is converted into glyceraldehyde will be converted into glyceraldehyde 3 phosphate, and this dihydroxyacetone phosphate will be converted into glyceraldehyde 3 phosphate. Now, fructose can be phosphorylated to fructose 6 phosphate by the hexokinase. Now, we know that this hexokinase is present in the liver, that is the enzyme which is also present in the glycolysis. However, what happens? The affinity of hexokinase for glucose is 20 times as great as it is for fructose. If we know that this hexokinase has the ability to convert fructose to fructose 6 phosphate, but the hexokinase has more love for glucose than for fructose. Therefore, whenever glucose is present, it will phosphorylate glucose rather than phosphorylating fructose. So, little fructose 6 phosphate is found in the liver because glucose is so much more abundant in this organ. We know that in the liver, glucose is present in abundant amount. Therefore, hexokinase has affinity for glucose rather than having affinity for fructose. Therefore, only a very little amount of fructose 6 phosphate, while glucose 6 phosphate is formed in a abundant amount. So, glucose as the preferred fuel is also trapped in the muscle by the hexokinase reaction. Now, because liver and the muscle phosphorylated glucose rather than fructose, adipose tissue is exposed to more fructose than glucose. So, what happens? This liver and muscle phosphorylates glucose rather than fructose. So, all the glucose is getting converted into glucose 6 phosphate. So what is remaining? Remaining is fructose. Therefore, in the adipose tissue, we find that more amount of fructose is present as compared to glucose. Hence, the formation of fructose 6 phosphate in the adipose tissue is not competitively inhibited by a biologically significant extent, and most of the fructose in adipose tissue is metabolized through fructose 6 phosphate. So this is the reason. Now, in this way, we have found that how the fructose enter into the glycolysis. So, remember that these three steps. Now, we will see the entry of the another sugar, the sugar of the milk, that is the lactose or the galactose. Now, this galactose is converted into glucose 6 phosphate again in the same way as the fructose. Fructose was converted into fructose 6 phosphate. Here, galactose is converted into glucose 6 phosphate in 4 steps. Now here it requires 4 steps. What are these steps? The first reaction is the phosphorylation of galactose to galactose 1-phosphate, again the kinase. The kinase is which kinase? Galactokinase. So galactose is converted into galactokinase, and the resultant compound which is formed is called as galactose 1-phosphate. So galactose enzyme galactokinase forms galactose 1-phosphate. In the previous example, in fructose, we found that fructose is converted by fructokinase into fructose 1-phosphate. Now here galactose 1-phosphate reacts with, now it reacts with UDP glucose. That is, it reacts with uridine diphosphate glucose. This UDP glucose and glucose 1-phosphate are formed. So what will be the resultant molecule? It will be UDP galactose and glucose 1-phosphate. So the enzyme is galactose 1-phosphate uridyl transferase. Why uridyl transferase? Because there is a transfer of uridyl derivative. Okay, so again the galactose moiety of UDP galactose is epimerized to glucose. So this galactose is now converted into glucose. The configuration of hydroxyl group at carbon 4 is inverted by UDP galactose 4 epimerase. So epimerization reaction takes place, and what we find is this galactose 1-phosphate plus UDP glucose we get, and this combines with uridine, and then this uridine, that is UDP galactose is there, and this is converted into galactose 1-phosphate uridyl transferase into UDP galactose and glucose 1-phosphate. So this is the UDP galactose, and a galactose 1-phosphate plus uridine means UDP galactose. This UDP galactose is epimerized to UDP glucose. This UDP galactose, and this is UDP glucose. So in this way this reaction takes place. So glucose 1-phosphate formed from galactose is isomerized to glucose 6-phosphate by phosphoglucomutase. So by the mutase reaction, this alpha D glucose 1-phosphate is converted into alpha glucose 6-phosphate. So in this way this can now enter into the glycolysis. So these are the reaction. In this galactose, we find that UDP comes into picture, so that it is converted into UDP galactose, and ultimately we get the formation of alpha D glucose 6-phosphate. So this is all about the entry of galactose. Then comes the entry of mannose. This is also very simple. Mannose is converted to fructose 6-phosphate in 2 steps. See, fructose was converted by 3 steps, galactose was converted by 4 steps, and this mannose is converted into fructose 6-phosphate in 2 steps. Hexokinase catalyzes the conversion of mannose into mannose 6-phosphate. Isomerase converts mannose 6-phosphate into fructose 6-phosphate. Now, fructose 6-phosphate is a metabolite of glycolysis direct, so there is no problem. So here what will happen? This mannose, by the activity of the enzyme hexokinase, will donate its phosphate group and will be converted to ADP. Mannose will become mannose 6-phosphate, and this mannose 6-phosphate will undergo isomerization and will form fructose 6-phosphate, and this fructose 6-phosphate will enter into the glycolysis. Now we will see some of the terms associated with these reactions. Number 1 is the intolerance to milk. Many people are unable to metabolize milk sugar lactose and experience gastrointestinal disturbance if they drink milk. Now this happens in many children. When after some time mother finds that the baby is not able to digest the milk. So this is the milk intolerance or lactose intolerance, or it is also called as hypolactasia. This is caused by deficiency of the enzyme lactase. This lactase cleaves glucose and galactose. Microorganism in the colon ferment undigested lactose to lactic acid, and lactic acid is very dangerous. This lactic acid is generating methane and hydrogen gas. So it will generate the to lactic acid generating methane and hydrogen gas. The gas produced creates uncomfortable feeling of gut distension and the annoying problem of flatulence. What happens here? The stomach of the small baby becomes very large. That is, it is called as flatulence. So the lactic acid is osmotically active and draws water into the intestine. This why the flatulence is there, because more amount of water is attracted because of the lactic acid. So more amount of water is there, as does in undigested lactose, resulting in diarrhea. The gas and diarrhea hinder the absorption of other nutrients, fats and protein. So what will be the treatment for this? To avoid the product containing lactose, the enzyme, this enzyme is given. What is the enzyme? The lactase. Okay, so we can give the lactase enzyme. Then comes the this is for the intolerance to milk, that is for the lactose. Now this is for the galactosemia. Disturbance of galactose metabolism is referred to as galactosemia. Classic galactosemia is an inherited deficiency in galactose 1 phosphate uridyl transferase activity. If this enzyme is absent, then that person will be suffering from galactosemia. What are the symptoms? The symptoms are vomiting, diarrhea after consuming milk, enlargement of the liver. Liver will this is called as hepatomegaly. So this liver will become large. Then jaundice will take place, sometimes cirrhosis and sometimes cataract. Lethargy and retarded mental development markedly elevated blood glucose level. So the absence of the transferase in blood cells is a definite diagnostic criteria. The most common treatment is to remove galactose and lactose from the diet. So the only treatment for these kind of children will be to remove the galactose from the diet as well as the lactose. That is, the person should be discontinued with the milk. So this is all about the uh, galactose, lactose, and mannose. Uh, these all the three types of sugars, they enter into the glycolysis, and in the after entering into the glycolysis, we get the entry of these sugars into the glycolysis. So after entering into the glycolysis, what happens? They now they are streamlined and they will lead to the formation of the pyruvate. This pyruvate will now produce acetyl CoA. This acetyl CoA will then combine with oxaloacetate to form citrate. Like this, the glycolysis will end up into this TCA cycle. So we know that TCA cycle is the major source of ATP for us. So this is all about the entry of sugars, that is fructose, galactose and mannose. In this way, we get the various sugars into the glycolysis. So if you have any questions, please ask. You can write in the comment box if you have any questions. Yes, please. If you have any questions, please write down in the comment box. Yes. Have you understood? Yes or no? This is very easy. You can very well come get the entry of these sugars into the glycolysis. So what I feel is this is all for today's lecture. Entry of glucose, mannose and entry of fructose, mannose and galactose. So the other things we will see tomorrow. Tomorrow also I will be taking the class, and in the class, actually because of the connectivity problem, I was not able to complete the quiz. But for today or by tomorrow afternoon, you will get the quiz as well as the assignment, and you will be able to solve that. So this is all for today. So thank you everyone.